and welcome to Season 7 of the Dementia Researcher iSTAART Relay podcast. In this series, members of the iSTAART's professional interest areas interview each other about their PIAs and the hot topics in their fields. Each guest then becomes the next episode's host, passing the conversation along from one researcher to the next. We're releasing one episode a day in the run-up to the Alzheimer's Association International Conference, this year in London and online, showcasing the work of the iSTAART PIAs. Thank you for listening.
Hello and welcome to Relay Live from Dementia Researcher and iSTAART. This session is part of Dementia Researcher and iSTAART's Relay series, which this year includes 6 podcast episodes and 2 special live streams. And across the series, we're meeting researchers involved in iSTAART's professional interest areas, or PIAs, as you're going to hear them referred to from now on, to learn about their work, the topics shaping their fields, and what the PIAs have planned around the AAIC, or Alzheimer's Association International Conference, which is starting in just over a week. I think we're about a week away from where the pre-conference activities start. So I'm Adam Smith from Dementia Researcher, and today I'm joined by Dr. Sam Lockhart from the Neuroimaging PIA and Dr. Ingrid Ekstrom from the Sensory Health and Cognition PIA. Hi, both of you. Thank you for joining.
Hey, thanks for having us.
Thanks for having us.
So, as I said, we're going to be talking about their research, the big questions in the field, the role of the PIAs, and what they're looking forward to at this year's AAIC. But Sam, I'm going to come to you first and ask you to properly introduce yourself and your research area and your role in the PIA. But don't jump ahead to answer my next questions. Just research field, your role in the PIA.
Sure. Well, hi, Adam. Hi, Ingrid. And hi, everyone. Thanks again for having us. So I'm Sam Lockhart. I am a neuroscientist and neuroimager by training, and I have a background in diseases of brain aging, cognition, and multimodal imaging biomarkers. I spent a lot of my career working in academic Alzheimer's disease research centers in the US, leading imaging components of various observational studies or prevention trials. But I now work in industry supporting imaging strategy and execution in various CNS clinical trials. And within the iSTAART Neuroimaging PIA, I serve currently as the programs chair, where I help to organize webinars and conference activities and awards and other opportunities for our neuroimaging community to help connect and collaborate amongst them.
Wonderful. And so I feel like neuroimaging is all about AI and machine learning now as well. Is that a hot thing for you?
It's definitely a set of hot topics, that's for sure. And I would surely have some opinions about their usage in various studies. For example, imaging generates a lot of data. There's a lot of predictive power that is available within it. However, there needs to be appropriate use of it as well. You know, prediction is only as good as it can also be explanatory as well, right? And so we have to figure out what are the best ways to leverage this data to actually try to figure out trajectories of late life brain health and who could actually be prevented from having certain diseases as well. And the right combinations of different types of imaging biomarkers becomes key there as well. We can start to bring in MRI and PET and SPECT and other fluid tools as well to try to characterize these individuals, and AI and machine learning are going to have key roles in that.
And I suppose as programs chair, you're one of the people or key people that gets to influence the programming of what the PIA is going to look at in those webinars. Is that something that you're doing all year round as well?
Absolutely. We're definitely doing that all year round. Of course, I would say our primary focus is around the Alzheimer's Imaging Consortium, which is a one-day conference that's going to be held next Saturday, the 11th, the day before the main conference starts. We have an entire day's worth of agenda with speakers, data blitzes, posters, panels, plenaries, and a fantastic year in review session that summarizes all the great imaging work. And then throughout the year, about every 4 to 6 weeks or so, we'll have some sort of educational or programmatic activity, whether that be a webinar to showcase recent controversies or updates in the field. We had one about tau PET harmonization methods a few months ago, to some primer series that we've been organizing lately, where we've been talking about like one specific methodology by the creators of that methodology, so they can come and talk to the field, to the imaging PIA and its membership about, you know, what was the background to it? How did they build this thing? And how are they trying to evolve this technology that they're building as well? We are constantly also on the lookout for any suggestions by our PIA membership, or anyone really, about other future educational activities or ways to try to engage our PIA membership.
Great. So busy job. But now I'm going to come to you, Ingrid, and ask you to introduce yourself, your research area as well, and what's your role in the sensory health and cognition peer?
Yeah, so thanks so much for having me, and hi Sam as well. So my name is Ingrid Ekstrom. I am based in Stockholm, Sweden at Karolinska Institutet at something called the Aging Research Center, which is a joint center between Karolinska Institutet and Stockholm University. And my background is in psychology. I spent my PhD in a psychophysics lab where I learned a lot about the basics of sensory processing and also neuropsychological assessments. And I am very much interested in particularly the sense of smell. So this is something that I'm trying to highlight in the PIA. And here at the Aging Research Center, I am assistant professor. I am trying to build my research group at the moment, and I'm very interested in especially looking into longitudinal trajectories of sensory decline and cognitive decline, and how they relate to dementia, normative aging, and so forth. And my role in the PIA has been the early career representative during the past years. But now we actually had a re-election, and now I'm the communications chair, and I'm eager to learn what this is going to entail. As an early career representative, I've been mainly working. To be fair, we are quite new PIA, so I've been kind of like the past years were the first years. So I've been really involved in building up the PIA and trying to recruit as many researchers as possible and try to increase our visibility.
I think you've got one of the most exciting jobs, I think, in all the peers, because sensory health particularly seems to be something that's been underexplored in the past and is finally getting the recognition as something that needs to be looked at more. So, so many people I've come across are really interested in that kind of how those sensors add into this. So you've got a lot to communicate, I guess. What kind of researchers tend to be involved in your peer? Are they clinicians? Are they...
This is actually what the thing I like the most about the PIA. They come from all types of backgrounds. So it's epidemiology, neuropsychology, but we also have ENT specialists and surgeons. And I would say that most of the PIA members have some kind of clinical background as well. I'm the only one who's not been working in a clinic, who's mostly focused on research. So it's a very mixed group of people, I would say.
Wonderful. But as we picked up on our last live stream and in the podcast we've released this week, is you don't have to work in sensory health to join a peer. You don't have to be working in neuroimaging to know the neuroimaging peer. You just have to have an interest, right? And that opens up this door to learning about something that's maybe a little bit beyond your field to make those connections to your own. Well, that's brilliant. Thank you so much, both of you, for joining us. And this gives us a really helpful starting point, because before we talk about the peers, I want to spend some time talking about your own research, because that's what often brings people into these communities as in the first place. So Ingrid, I'm going to come back to you to tell us more about your own research and what you're currently working on.
Yeah, I thank you so much. I really loved also thinking about this question, because quite often you're so caught up in your work that you kind of forget what it is you're actually doing and why. So I'm happy to discuss this. So I already said that my biggest interest is in the sense of smell. And I think it's mostly what attracted me the first place was that it's a very underexplored sensory system as compared to vision and hearing, especially. And I have been mostly working on, in my PhD, I've been trying to find out if we can use olfactory measures to predict dementia occurrence in population-based studies, also in combination with other markers and mainly cognitive markers, but I'm also really interested in neuroimaging, actually. So this is something that I have been eager to learn more about during the past years as well. And right now, I'm mostly using large-scale population-based data to see how we can best predict dementia occurrence based on olfactory markers. But I'm also very interested in how sensory decline develops over time in old adulthood, and increasingly interested actually to have a more kind of a life course perspective to it, because I think 10 years ago when I started looking at this, we knew hardly anything about the sense of smell in old age. And now we actually have the same situation. We don't know anything about the sense of smell in early stages of the life course. And I think it'd be really great to kind of like bring this together. So yeah, and I'm also really interested in kind of like just to look at more like the mechanistic component of these questions. So like, why is it that we can see that especially olfactory markers are very much, yeah, are predictive of dementia occurrence quite early, and how we can use that information to ideally improve dementia detection.
Do we have a sense of what percentage of people who go on to be diagnosed with dementia experience one of these sensory losses, whether it be that taste or smell or other things?
I mean, of course, there's like a lot of variability between studies, but some studies would say that almost all, like 99%, some would say 80%. So anything between like 60 and 90%, I would say, depending on the measurement.
And is that likely? I mean, does that come before we see cognitive changes?
Yes.
Generally?
Yeah, we can see it already in people who don't have any like cognitive symptoms, and quite often like several years to decades before.
That's fascinating. And because I was reading an article the other day about smell tests, which I was just intrigued about how you test for this. What are the kind of strengths and limitations as those kind of common smell tests for odor identification and things? How might you? Because that got me into thinking about how you might apply that test in a healthcare setting early on to add into this.
Yeah, I think like the biggest strength of these tests is that they're really easy to assess. So you basically have these pens that they just look like markers, like usual markers, and you open them and you present them to participant, and they just smell like common household odors. And then in the identification task, you ask the participant to name the odor. But then I think like the biggest challenge here, like although it's a very simple test, it doesn't take a lot of time, it's quite agreeable to most people, is to kind of produce smells that actually really match what an odor really smells like. So because you usually don't present like the real household odor, you don't present a banana, you don't present like an apple or a rose, because that wouldn't be standardized. So you really need to kind of make sure that these molecules are similar and like they evoke the same smell. And then, yeah, sorry.
No, carry on. Sorry.
Yeah, I think the biggest criticism has always been that we often use this identification task so we ask people, what is this smell? And some people say, but this is like carries a heavy cognitive component because of course, there's a verbal aspect to it. Which is actually not a problem, because when we actually compare these measures that are having this verbal component incorporated with more like threshold-based measures, we can actually see that it is actually the ability to identify odors correctly that is a very good marker for dementia. So it is not only kind of like a sensory impairment, but also more kind of like a integration with language networks in the brain and memory networks that is giving us this great signal for dementia. So I would say that that's kind of like the biggest criticism that I think is also kind of like a strength. Yeah.
You can see how that's a problem, isn't it? Is even if they could identify the smell, the word finding and the ability to remember that is a burning smell, or that is fresh cut grass, or whatever it is, that finding that. Do people misidentify smells as well? Do they think that something smells that's not the right thing?
Yeah, so that's actually something, and it's not even kind of like a misidentification. It's mostly that the smell is actually really recoded differently. And that has actually happened quite a lot after the pandemic for some reason. And it had actually devastating effects on some people's lives. Because smells that we usually think are quite agreeable, like fresh buns from the bakery, or chocolate smell, or other like common household odors that are pleasant, they suddenly would carry like a rotten characteristic. And that was really, really bad for the people who are affected by it. But that's actually something that we don't really see in relation to dementia.
That's fascinating, isn't it? I mean, you can see how that can also then cause risk, that if people can't identify the smell of something burning, or we rely upon those sensors, don't we? In that, I mean, obviously we're focusing on smell here because that's your big thing as well. But do you get the same? Is it the same in taste as well?
Yeah, I mean, I guess the really interesting thing with smell and taste is that like a big portion of what we commonly describe as taste is actually smell. So you can actually test this with yourself. You can try to basically just like try to hold your nose and really close it and try to like chew, let's say like a jelly bean, and they all have different flavors, right? But if you basically block the nose, they all taste the same. So a lot of the taste experience that we have is actually smell. And given that there's this big overlap, it's very different to kind of like separate the two. And we see that like taste is quite often intact in aging and dementia as well. It's mostly like kind of like the information that comes from the olfactory system that is affected.
In Tuesday's show, we were talking about sex differences. Are there sex differences in sensory health as well?
Yeah, I mean, this has been actually researched quite a bit, because we can quite often see that women outperform men, that they have a better sense of smell, and that they are commonly also more interested in olfactory cues and have like a nice smelling environment and so forth. I don't know if you can relate to that in your own family, but I can definitely do that in my household. But then we could also see like there's a big meta-analysis that has been done like a few, I think maybe 2 years ago, where they saw that there are sex differences, but they're quite small. So, and there's also no difference in rate of decline. So women and men, they decline in parallel as they age. There's no like accelerated decline for men.
And so coming back to your research, you're particularly interested in looking at this and how it's an identifier of future risk, rather than trying to address the symptom, which is to kind of give people back their sense of smell or their sense of taste.
No, actually, I just kind of like forgot to mention that aspect of my research. So during my PhD, I was really interested in olfactory training, how to improve the sense of smell. And we've done a few studies about that. And more recently, actually, the I guess like the direction that I'm taking with my research is more and more to understand what would be like a consequence of smell loss, because we don't really know so much about that. And try to identify also, I mean, now we know already that it is a marker for dementia. We know that a lot of people who are living with dementia have a reduced sense of smell, but not only dementia, it's also other very common diseases in older age. So what is the consequence for these people? And is there anything we can do about that? So I'm definitely interested in that question. I would say across the lifespan. I'm planning for a study on children born prematurely now, to see if there's any effect of premature birth on olfaction, and to see if that actually has some impact on their quality of life. And
you can see how addressing the symptom is important, can't you? I know obviously when people have primary progressive aphasia or one of the aphasias, that having access to a speech and language therapist to try and enable you to continue to communicate is important. And if anti-amyloid therapies are going to do well at giving people longer with, some form of dementia or decline, that doing things like restoring senses, which are what make you happy in life, right? I mean, your ability to smell things and taste food and go outdoors, and they're important things to quality of life. So it's important that we don't just say, oh, well, that's it, you've lost your sense of smell now. Yeah, that will never come back. If there's things you can do to address that, it feels really important.
Yeah, I'm very happy you say that, because when I started doing this type of research a few years ago, people would kind of like laugh it off a bit, like, oh, you look at the sense of smell, that's kind of cute. Who cares, really? And then when I would mention, yeah, but it's an early sign of dementia, then it was given some weight. Then we're like, yeah, of course. But yeah, it's one of our senses. And I think that during the pandemic, people actually realized how much it is important for them. And it's not only enjoyment of food or navigating the environment, it's also a very, very important aspect of attachment between children and parents, or but also to your partner. And I think the interesting part is that it's actually only noticed that when it's gone. So, and quite often, if you lose your sense of smell gradually, you will not really get so much aware of it. So you will kind of like feel like something is off all the time. Food doesn't taste anymore, and so forth, but you will maybe not notice it so much. But if you lose it very, very sudden, like during COVID, most people suffer from it.
And is this something that's a bit like hearing loss, where we know that people's hearing loss can decline over a very long time, but the brain is really great at compensating, and so, as you say, you don't really realize it's gone? Is it the same with taste and smell and other things?
That's actually something we don't really know, because there are not so many studies that have tracked people and started early enough. What we can see in our data when we start looking at 60-year-olds, we can see that the people who experience the greatest olfactory loss over time, they had already a very bad sense of smell at the beginning. So we actually need to track it earlier, I would say, to answer that.
I always remember somebody I met very early on in my time in working in dementia telling me how one of the things that had made her most sad about getting developing dementia was that she couldn't drink tea anymore. It tasted, as you say, she absolutely hated the taste of tea. She'd have been a tea drinker all her life. She'd, her 5 cups of tea a day, and could no longer drink tea. And that's one of the things that she kind of missed more than many other things. So thank you very much, Ingrid. That was fascinating introduction. Sam, you've been very patient, but I'm going to come to you now. So from your perspective, what are you working on right now? What's your research around?
Thanks for asking. Let me also say that entire conversation was fascinating. I've long also been interested in the sensory and motor integration into cognition and the function of brain networks in, but not only processing that information, but being early prognostic indicators of decline in patients with dementia. So that's fascinating. So at its broadest level, I guess my work has been involved in using imaging to try to understand brain health and brain disease in a way that is biologically meaningful and hopefully clinically useful. So earlier in my career, in the academic phase of my life, if you will, that meant studying how things like vascular and metabolic risk factors, or things like amyloid and tau pathology, or changes in brain health like atrophy as measured using MR, or cerebral perfusion as measured using cerebral blood flow imaging, trying to understand how these imaging markers could relate to cognitive health and brain health, and particularly in what is often termed a preclinical phase of decline, so before the stages of dementia, when individuals are otherwise ostensibly cognitively normal. But recently, as I've actually shifted toward industry work, a lot of my attention, my interest have shifted toward things like, how do we bring imaging biomarkers, particularly brain imaging biomarkers, responsibly into a clinical trial settings? So how do we do things like standardize our acquisition across different sites and countries and different types of technologies? How do we actually develop imaging endpoints that we can interpret in a study to try to track the efficacy of some sort of therapeutic, for example? How do we manage the operations of that sort of thing? Bringing in scans and presenting them to neuroradiologists to read for amyloid positivity, and to imaging pipelines to quantify them for things like amyloid centiloids, is fairly straightforward on its surface, but when you have to make the operational decisions of how that's going to be played out, there's a lot of scientific considerations in that. So I find I'm actually using the same scientific muscles that I used in academia now in a clinical trials setting for a company that's what's called a CRO, or a clinical research organization. We help to run the clinical trials on behalf of the companies that are trying to test different therapeutic agents to try to get them for approval into the market. And so what I'm trying to understand is how we bring in this operational aspect of how you run these studies with these clinical sites, these imaging sites all over the world, and how you can then generate this imaging data that is, again, biologically meaningful and might have actually some value to the clinical interpretation of who these patients are and how they might be responding to different types of interventions. And in particular, you have to make sure that imaging can add value in that setting rather than just unnecessarily adding complexity or cost, for example. So I'm really interested in this space now where we try to bridge things like biology, but also patient selection, precision personalized medicine type of considerations, and do that in a setting of disease monitoring, but also looking at things like treatment response.
And so what's one of the biggest barriers you face in trying to bring that about?
Well, I mean, let's think about it.
That there's multiple
conversations. Yes, I mean, where to start, and they're all present, all that you can think of, and they all have to be thought of relatively simultaneously. But everything kind of starts with a clinical trial protocol, right? We have a drug that's being tested for a certain indication in a certain type of patient population. Let's just pick one of the most obvious ones, things like monoclonal antibodies for amyloid removal in the setting of mild to moderate dementia. The goal is to try to assess people who have some degree of brain amyloid on the brain, So as measured using something like PET, so that you can then recruit the right patients in that have something that the drug is trying to target. And then you actually need to monitor them successfully longitudinally over time to be able to detect not only a difference in the patients on drug, obviously, but a difference in that slope relative to the placebo group. That also has to be combined with things like MRIs to look at eligibility, to make sure you're recruiting the right patients who don't have certain inclusion and exclusion criteria, and also to track them for safety over time, because these drugs have certain safety risks, things like ARIA, that need to be monitored as well. And so you're quickly then bringing in multiple different modalities that require multiple different imaging facilities, that require multiple different things like PET tracers that have to be navigated around from country to country or from site to site. And certain things are just not available in certain places of the world, right? Like the infrastructure that is necessary to run certain things like these big phase 2 and phase 3 clinical trials is fairly well established in areas like North America, Western Europe, Eastern Asia, and parts of Australia, for example. But there's a lot of growth that needs to happen to build this infrastructure in areas like Sub-Saharan Africa, Middle East, Central and Eastern Europe, Latin America. And so there's a lot of places that studies just plain can't be run right now, or can't be run without a lot of investment or changes to the infrastructure. And so I think, if I will, the ability to then generate data sets from clinical trials that have representative and diverse populations, so that we can show that these drugs are actually being tested in a broad swath of individuals who can get to the representative of who can get the disease, and that we can actually monitor and see the same types of effects in these populations, really cannot be done that very well right now. And so there needs to be a lot of investment from various countries, various pharma companies, and various other players in this space to sort of build up this type of infrastructure. Because again, once you start thinking, I need to get 3T MRIs, I need to get next generation PET scanners, I need to get regulatory approved amyloid PET tracers, and I need clinicians who can assess people for the presence of things like dementia symptoms and cognitive impairment. There's a lot of interfacing things that have to come together there. And we are still building those things in quite a few places in the world.
And is this where, I mean, we touched upon it at the very start, this is where you could see the technology, and which is so rapidly progressing in your field and in, in all fields, let's face it, could really come to play here, where could technology be used to level that playing field, so it can take the outputs and the scans from different equipment, different quality equipment, using different things to try and then standardize that using artificial intelligence and things. So you can do this without having the multi-million-dollar equipment in every clinic.
These things are definitely being explored, and they are probably a very valuable pathway toward achieving those types of goals. For example, a standard MRI scanner, the comment is usually it costs about $1 million per Tesla to get one of these things installed. So we're talking several $1 million for the installation of a thing like a 3T MRI. But there are these lower field strength MRI scanners that have been developed and are starting to be implemented in certain parts of the world. They generate lower resolution, if you will, lower quality images, but some of these companies have also developed AI tools to effectively upsample or use AI to sort of back interpret some of the data that can be acquired from some of these scanners. And some of them are starting to generate data that suggests that they may be able to have quality data to do things like scanning people to determine whether they have certain exclusion criteria like strokes already on the brain or edema, and look for the presence of things like microhemorrhages longitudinally over time, which are one of the key safety things that we're trying to look for in something like a monoclonal antibody therapy. But I would say that those things, while they are being explored and people are trying to establish their validity, I would say the field is still trying to assess whether they are comfortable with using those in something like a large-scale clinical trial. One of the things that I've noticed is that you sort of need to go with a lowest common denominator approach, or you want to have a relatively similar playing field for all of the tools that you're trying to assess out there. And so if you're going to have things like higher quality PET scanners in North America and Europe, but try to mix in something like an ultra low dose PET or a low field strength MRI scanner in another part of the world, how are you going to integrate those data sets together to be able to interpret those patients with respect to one another as you put them together for your statistical analysis? And so that presents some risk. And again, a company that might be trying to develop one of these assets might think that that is too much of a risk, and it is a less risky decision to avoid that entirely, not go for some of these regions where you might need to bring in something like a low field strength MRI and AI upsampling of the data, and just go with another approach, which is use our historically established things like 3T 1.5 MRI scanners or current generation PET scanners and things of the sort. So it's kind of a double-edged sword, and there's always a give and take there. But money talks as well, right? And the cost of these things is going up so much that I feel like the field is going to have to move in this direction, not only to expand representativeness of patients that are in these studies and who actually is being assessed for the quality of these drugs, but it will hopefully lower the cost of various companies running these studies and various companies getting into this space as well to test their therapeutics. So I think there's going to be an explosive growth of this in the next coming years is my long story.
Yeah, I can see that, because it seems unlikely that any time, I suppose it's a tough sell, isn't it, to persuade companies to invest in those countries to put that hardware in place in the first place when it's already exists in other places. But if they really want to address, some of these gaps, it has to be done.
Yeah.
And
then if you're talking about mixing that with PET, for example, you have to talk about PET tracers that are coming in as well. I'll make one last pitch that we actually have a plenary at our imaging day on this very topic, on ultra low field and ultra low dose scanning for scalable neuroimaging studies.
Well, I was going to say that on PET, because obviously we can talk about MRI, which is obviously that much easier. And as you said, there's the low field elements coming in now. But PETs is a whole different thing. Again, I mean, we haven't even got PET scanners in parts of the UK, never mind in parts of sub-Saharan Africa. And then you've got the maintenance. It's not just like it's the $3 million to put in a 3 Tesla MRI. You've got to sustain that piece of kit then, haven't you? I've certainly read a few articles recently on Nature where they were talking about expensive kit that got installed and now has just been sat there for a year, 2 years, not used because it broke down and they can't get it fixed, which seems like waste of the investment in the first place. But that's not all your work, of course, Sam. So you're kind of, I think the exciting bit there about bringing that consistency is quite exciting too.
Yes, and what's also interesting is that even though I began my field more in Alzheimer's and related conditions, once you start taking imaging tools and try to utilize them in a clinical trial setting, I don't want to oversimplify it, but an MRI scan is an MRI scan, a PET scan is a PET scan, and we can start to look into different brain health areas and different disease indications that also might be trying to use these technologies, because as long as the company that's testing the therapeutic agent wants to look at something like strokes on an MRI or atrophy on an MRI or changes in tracer uptake on a PET or a SPECT scan, we know how to do that. And so we've been getting more involved in studies like Parkinson's research, which has shown an increasing growth in people trying to test various therapeutics and also using brain imaging as well to try to assess people for eligibility and safety and efficacy, as I've been saying. Indeed, there are some overlapping conditions like dementia with Lewy bodies where we might actually be able to test some of the very same therapeutics being looked at in Alzheimer's disease in this population that is sort of if you will, a midpoint, not really, but a midpoint between things like Parkinson's and Alzheimer's disease. Can you clear out the brain amyloid to try to treat at least some portion of what is the disease pathology that some of these patients are experiencing? So it's a fascinating time because it's also allowing me to sort of expand beyond that background I had in Alzheimer's disease, particularly to more broadly understand diseases that affect people as their brains get older, which of course become comorbid as well. We start to accumulate more and more of these in combination statistically as a population. And so it's really fascinating because I've also then been able to sort of somehow get closer to the treatment of patients, even though, of course, I'm not a clinician, right? I do feel like I have a closer feel on the pulse of what are the questions and what are the ways that we're trying to, as a field and as a collection of various pharmaceutical and biotech companies out there, trying to develop drugs that might be able, drugs or other interventions that might be able to actually bend those curves and change late life brain health.
And I'm going to be slightly tongue-in-cheek when I say I think neuroimaging stood a great fight back as well. I remember, I'm sure it was only 4 or 5 years ago, seeing Nick Fox at the AAIC talking about almost making the case for why neuroimaging was still relevant in a time when fluid and blood-based biomarkers were seemingly the panacea. This was the future. But I think neuroimaging's done really well to maintain that it's still absolutely essential despite the progress made in fluid biomarkers.
Yeah, lot of thoughts on this, obviously. And as somebody who does imaging as my day job, right, there's a certain amount of vested bias I have in this. But actually, people who I talk to a lot will know I say this, right? Like, I know imaging only has a certain amount of half-life left in terms of its relevance, because there's going to be better markers that are peripherally derived markers that are going to come along and sort of be less expensive or less invasive or more expansive in their reach. And are going to be able to sort of take the jobs of certain imaging biomarkers bit by bit over time. And we can take amyloid PET as, for example, for one of these. 15, 20 years ago, before we really had amyloid PET commonplace, we were recruiting patients who didn't even have brain amyloid into Alzheimer's studies, testing for anti-amyloid therapeutics. That was, of course, a mistake. Then we started to get amyloid PET, where we could actually say, we're certain they have amyloid on the brain, to be able to then target that and try to remove that. And we were successfully, again, as a field, able to show clearance of brain amyloid through things like amyloid PET scans. We are now getting better able to use certain fluid biomarkers. I'll pick p-tau217 as one of them, that is very, very effective at characterizing the level of brain amyloid on an individual, at least at a single point in time. So you can recruit people for eligibility into a study now. If all you're interested in is asking the question, do they have amyloid on the brain? A p-tau217 plasma blood test is probably going to do just as good a job as an amyloid PET scan is, or at least about 90% as effective, and probably be a lot cheaper and more scalable. However, we're still trying to figure out how well do those changes in p-tau217 plasma measurements track over time, and how well do they actually track the efficacy of the drug that's being tested? Are there other comorbidities or considerations that need to be explored to understand how to interpret those longitudinal measures? So, for example, amyloid PET's now in this place where if you just want to look at baseline, you might be able to use a plasma measurement and do the job for a fraction of the cost. But if you want to look at changes in brain amyloid, I would say jury's still out, and you do still kind of need amyloid PET for that question. Tau PET's a few years behind that as well. There's a lot of growing evidence in this case, and there'll be some presented next week at AAIC, but I would say we don't have a solid case for a tau fluid biomarker that replicates what is seen on tau PET. And furthermore, one that tracks it longitudinally as well. And so in the absence of that, something like tau PET to characterize presence of tau on the brain and changes in tau pathology, either a stop of the spread or a reduction of tau pathology, imaging is the tool that you're going to need for that. And of course, imaging also presents some spatial information about where in the brain these things occur that a plasma measurement or even a CNS-derived measurement like cerebrospinal fluid measurement cannot provide. And there are no, as far as I know, good peripheral markers for tracking things like ARIA on MRI, or understanding certain other characteristics of MRI atrophy or neurodegeneration. We have some tools like neurofilament light, GFAP, and other measurements that can try to track that. But I would say that, yes, imaging still has some place in this question and will continue to have some place in this question. But bit by bit, other tools are going to come and sort of knock them off the pedestal, or at least make them the second best option or something like that.
It's okay, Sam.
That being said,
I'm sold. I'm sold. I buy it. You can keep. You can stay. No. I was teasing. But you're absolutely right. You can see it. Neuroimaging is not going anywhere anytime soon, is it? And it's still
well. And then there's some targets we don't have either, right? Like for alpha-synuclein, we don't really have a good brain imaging target. We don't really have good fluid targets. We have some that can characterize presence of pathology, but not really a continuous amount of them on fluid or tissue markers. And so it's sort of a race to the finish line for something like alpha-synuclein pathology. Who's gonna get there first? Imaging, biofluids, tissue? Are they going to have to come together in some fashion to better characterize that? And again, it's not just conditions like Parkinson's that get alpha-synuclein, but other related disorders and even comorbidities, copathology in patients with Alzheimer's.
Yeah, and we've seen other exciting areas like the retinal eye scanning to find things, and there's lots of exciting spaces. Brilliant. Well, that's a really useful setup for the next part of our conversation, where I want you to step back a little bit. I think, Sam, you might have already answered this question a little bit, but we step back from your own field to turn to some of the bigger debates that are happening in your wider fields right now. So, Ingrid, I'm going to come back to you and say, what are the really hot topics in sensory health and cognition research at the moment?
So I would say that there are three different ways to answer that. But when it comes to especially the kind of like the marker field, I would say, like why is it that we can see that the sensory health can be used as a marker for dementia? I would say that we really move much more into like mechanistic studies, and I am really excited about that, because it's like now I think we have the epidemiological studies really seeing like a quite large association and great predictive value. But what are the mechanisms? And I think that they're actually neuroimaging studies are really important for that. We're much better in also integrating olfactory stimuli in the scanner, so we can actually measure that. And if I would say that, just kind of like moving away from olfaction now, of course, I would say that the whole kind of like focus is on hearing now, with finger, like the finger study, also incorporating hearing now, and we have the Lancet Commission who published these very influential papers. So everyone is very interested in hearing, and if it is actually a potentially modifiable risk factor. I would say that that's kind of like, and then I would say the third one is moving a bit away from starting to look at sensory health only in older age, and starting to much more kind of like see how we can use already midlife sensory impairment as a risk factor. There was one very influential study, I would say, published in Alzheimer's and Dementia by Natasha Merten and her team this year, where they saw that if they looked at a lot of different risk factors or potential risk factors, everything from genetics to biomarkers to lifestyle factors, demographics, olfactory testing was one of the best predictor already in midlife. So I would say that's kind of like the third hottest, according to me. I would say that we kind of like move away from just testing it in old age and already like start in midlife, or what I'm kind of like increasingly seeing as young adulthood.
And is there a particular area where you feel that there needs to be stronger evidence? Is it that midlife place that you were talking about?
Yeah, definitely. We have very, very little studies that even have tracked sensory function during lifespan, and compared it to cognitive trajectories. And I would also say that we need better neuropsychological measurements. So because we in the PI are very interested in the interface between sensory function and cognition. And I would say that kind of like trying to improve the cognitive batteries that we use nowadays, they're kind of, I would say that like the real life component there is sometimes missing. And we have like a lot of like directions now in this type of research where we look at like different app-based cognitive testing and how to kind of like make it more adapted to also, I would say the generations that are about to age, which is Gen X and some kind of millennials also soon. So,
and is yours a space that's embracing technology as much as they are in neuroimaging there? Because as you touched on wearables there, and I know in the technology peer we did on Monday, they were talking a lot about nearables. Is this a space that's getting embraced?
I mean, definitely. And I think it's also coming a lot from like outside academia, I would say. I mean, there are a lot of like startups that are really interested in developing devices and apps. And I would say that like academia is moving into the more like naturalistic setting direction together with different companies outside academia. So I think there's a lot of things happening there. I don't want to claim that I really know so much about it, because I'm very, very much focused on academia. But I would say that there are a lot of things happening there.
So we did a podcast earlier in the year on XR and spatial technologies and things, and some of the augmented reality tools that people have come up with to augment existing reality around the house with everyday things where there'd been some kind of sensory loss could compensate for it. So, for example, putting red flashing lights on the stove to show that this is going to be hot, or putting something down the stairs to show that if you'd lost some of your depth perception, that actually having an augmented reality tool built into your glasses could help you still cope with, stairs and things that you might do to live at your own home for longer, which I thought was really exciting. It's not probably in your same space, but
no, but I mean, we have attracted some people in the PIA also. We have attracted architects, for instance. We had like a submission now for the FRS that was not accepted, unfortunately, but we actually had an architect kind of like looking into how we can how we can design living spaces for people with dementia with sensory specific factors in mind. So I think there's definitely a lot of things there happening as well.
Yeah, well, that's important, isn't it? And you see lots of nursing homes talk about their sensory gardens. I mean, usually, but when they often think when they talk about that, they're more in mind of dealing with autism and some of the other neurodivergent issues rather than particularly with dementia in mind. But, you know, they're important too. And
I definitely, I think like one reason for that is that still I would think that a lot of people who work with patients with dementia are not aware that for instance, that sensory issues are common. Some of them who have worked with these patients for a long time, they might kind of like have noticed this through their own experience. And then when I give a lecture and they come up to me like, oh, I've always knew this, but like I would say that we have within this field, it's like so new, so it has not really reached kind of like common knowledge, and not even with people like working with people who have dementia. So it's something that they almost get like an aha moment where they say like, of course, this is why they don't have appetite. This is why they always say that the food doesn't taste like anything. This is why we sometimes smuggle in this kind of like, I don't know how you say that in English, but we have muggy in Germany and in Sweden. It's this kind of like glutamate taste enhancers that some people say are really, really unhealthy, which they aren't actually. So it's actually better to enhance your flavor of your food with that than not eating at all.
Yeah. See, we've done a couple of talks before as well with Elise Parkinson looking at hydration and how important that is, and how, you know, all these messages saying, oh, you shouldn't drink coffee and tea, which is absolute nonsense. All fluids are good fluids, and I don't know if all food is good food, but I get where you're coming from.
I'm not sure all fluids are good fluids. If you think about dehydration.
Okay. Yeah, fair enough. Yeah, but that's fascinating. Thank you, Ingrid. And Sam, I'm going to come to you. What's the hottest topic in neuroimaging right now?
Oh, hottest. Well, let's see. I would say in particular, let me harmonize on what I was saying before. I think, how do we move from these very technically impressive imaging tools and results that we're getting to actually getting something that is interpretable or reproducible or valuable in a real world setting? Because we have these really sophisticated scanners, tools. We can acquire process images at a level that was unheard of 5 or 10 or 20 years ago. But now these harder questions kind of sit at the intersection of biology and translation of what that means. So how can we interpret what we're actually measuring in a biologically meaningful way? Can we demonstrate that these changes in a trial setting that these changes in an academic setting will work in a clinical trial setting or in a clinic with a real patient in front of you, for example? Can these highly controlled research environment data remain useful in diverse populations and sites or in healthcare systems across various countries? So it's not, I think, just necessarily about collecting better data, but it's about turning these measurements into knowledge that is reliable and helpful in improving the scientific and clinical decisions that we make. Whenever I'm reading a paper now or reading results that are put out there, it might seem like a striking, technically impressive result. But my first question, is now trending more not toward, is it statistically significant, but will this actually be true, say, in a different cohort or on a different scanner or in a different population? Or does this difference that's being measured actually have some clinical import? Because if the answer to those is yes, then I think we're much closer to using imaging as something that's actually genuinely useful. And so I hope, and I do think that more researchers are finding themselves asking, what do these imaging findings mean, not just in isolation, but what do they mean for cognition, daily functioning that people exhibit, and their quality of life? It's not just about what we can measure, but how can we integrate these imaging tools with all of the other tools we have, plasma and fluid biomarkers, sensory health measurements, other types of more sensitive cognitive assessments to get a richer picture. And until we can really get more successful at that, I think we're going to need to be better about developing those so that we're actually studying what we can do to help people's lives and not just develop better imaging endpoints.
And is there more that the research community could do to enable that? Because I feel that particularly when you get industry involved, that kind of element of competition and secrecy comes into play, that where you're talking about sharing methodologies that will enable people to be a bit more standardized, to look a little bit more holistically about how a trial was delivered, that could be a barrier to progressing what is clearly a challenging area.
Yeah, it's an expensive line of work, right? And so collecting all of this imaging data in an academic setting is very expensive, millions of dollars a year to collect PET scans, even at a single center, longitudinally in a cohort. And so, especially if you're going to want to track, a long-term response or a response to some type of intervention, getting availability of clinical trial data, and particularly clinical trial imaging data, will become very useful. And there are growing numbers of consortia out there, or sort of industry academic partnerships, that are starting to make more of these things available. Now, they tend to be more from, say, the placebo-controlled arms of studies, or from prior studies that had therapeutics that failed or did not move on to further phases as well. And that's a fairly good start, right? And so that's going to allow anyone out there in the field, industry scientist, academic scientist, to actually leverage more, if you will, real world clinical trials data that not just has imaging, but other biomarkers, other clinical and cognitive assessments on real patients who are actually being treated and tested for therapeutic efficacy of a drug. And that we can all start to work from those common and shared data sets from various trials to better understand and interpret what is a biologically meaningful change. Or let's flip that on its head. Who is the right patient at baseline that you want to recruit into a study? There's likely data out there from some of these clinical trials that says this drug may not have been successful overall, but in a subset of participants it was. Now, that's not great for the people who were testing it, because they had to a priori ask a question differently. But if you can start to figure out, well, gosh, if they had only recruited this third of people based on this set of characteristics and tested a larger set of them with a different primary endpoint, they would have been more successful in testing this very same therapeutic, or a therapeutic that like that. So how can we better identify individuals who are at risk for decline, but also at most therapeutic potential for certain types of assets out there? And then, again, turning questions around here, how do you best measure that longitudinally over time? Is imaging the only or one of the ways that you want to try to characterize those changes? And so that's, I think, where we're going to start to see a lot of exciting growth in coming years too.
Wonderful. Well, you've both got some challenges ahead for you, but it sounds like an exciting time in both fields. We've got 10 minutes left, and we really need to talk about iSTAART and your plans for AAIC. I'm going to come back to you, Ingrid. Given we've only got 10 minutes left, I'd like your big pitch for the Sensory and Health and Cognition peer. Tell us, what do you get up to, and why should people come along and join your peer?
I mean, first of all, I'm really good in pitching things, but I will try my best. So definitely, it's a very, I would say, given that we are very new peer, there's a lot of enthusiasm, and they're actually genuinely nice people. So what I've been enjoying most, actually, the past 3 years, have been like the meetings with my peer colleagues. They've been kind of like following me through like a lot of different things, academically and personally also. And I think it's also because it is a very multidisciplinary peer. And I mean, if you think about it, I mean, you cannot say that you're not interested in the senses, right? I mean, I would say that they're kind of like, it's the most fundamental thing for experiencing the world. I mean, it's even becoming like before cognition. So, I mean, get curious about it and what we do. And I think that it's also kind of like, if you want to join a peer where you really feel like you can build something, then I think it is the sensory peer. So
what have been some of the kind of most exciting outputs in the last year? Have you had some papers or some collaborations, some guidance? What
yeah, I mean, we're about to submit our first kind of like a narrative review, so like a position paper from the PIA that I've been working on a lot with the PIA. And I don't know, I've been thinking also that like different webinars where we really feel like we had like a very great outreach when it comes to kind of like getting to know the basics about sensory science. And I have a feeling that there's an increase in interest. And I would say that's kind of like the best thing we have done so far, to also attract a lot of younger students and early career researchers. So I've had actually through the PIA, I had like a lot of students reaching out to me wanting to come to my lab, and I'm really, really grateful for that. I can really see that there's a new generation being very excited about this.
That's wonderful, and I agree. I think I'm going to make the argument, the pitch for the peers peer, which I helped set up and was the first chair of, and certainly the sensory health one feels like everybody could be in those peers. I mean, okay, there are others in there as well. The aware peer and things like that doing great work, of course. But you're for everybody. Is the neuroimaging peer for everybody as well, Sam? Would you make that argument? Give us your pitch for neuroimaging. Here's your 2 minutes on neuroimaging peer.
I would, and let me also caveat that I'm one of the types of people who does sign up for every PIA because I'm interested in all of these things and want to stay abreast of all of the work, which is sometimes a little much. But the imaging PIA is a community for, as we've been describing, not only people who use, develop, and interpret imaging biomarkers, but also those who simply want to understand brain imaging and Alzheimer's and related dementias a little bit more. That can be methodological folks, analysis folks, people who are developing computational approaches, clinicians who are interested in translation, or really anyone interested in those areas, or also, as I've been mentioning, the intersection with things like fluid biomarkers, genomics, therapy development. You do not have to be a technical imaging specialist to come and belong in the imaging PIA. We do need people like clinicians, basic scientists, trialists, statisticians, trainees, and people who are just interested in thinking about things like imaging implementation and equity. And I think the PIA tries to create a space for all of those groups to talk together. We are looking to bring together people from different backgrounds who have this common theme of wanting to talk about how better to acquire, process, measure, and interpret imaging tools, particularly brain imaging tools.
And is I can't remember the stats, but is the neuroimaging the biggest PIA? Have you got the most members?
I do believe we may be the oldest and maybe the biggest PIA, and thanks to that vaunted position, we also have that entire day-long imaging pre-conference that has been established.
Why don't you go on and why don't you carry on and tell us what does the neuroimaging PIA have going on at AAIC this year?
Mm-hmm. Yes, to also answer your first question, last I saw, I think we had well over 1,700 members in this PIA across many different countries and continents as well. And we're always trying to figure out ways to better engage those individuals who may not have the ability to travel to where our conferences are usually, which is North America and Western Europe as well. But we do host every year the Alzheimer's Imaging Consortium on the Saturday before AAIC, which is a day-long, single-stream, one-room type of sessions where we'll do things like have an annual year in review talk by a trainee who summarizes a lot of the newest research presented in imaging and Alzheimer's and related disorders. This year, Luigi Lorenzini is presenting on that. And actually, he's turning that into a white paper submission that he's actually just submitted to a journal yesterday. So one of our productivity outcomes as well. We'll have things like sessions on tau PET advances, treatment effect and prognosis, the ultra low field and ultra low dose imaging that I talked about from our plenary, poster sessions where we'll simultaneously be hosting mentoring conversations at the tables during the poster and lunch sessions. We'll actually have a panel from radiologists and clinical experts and imaging trialists who are going to sit together and talk about how we're going to use things like MRI for ARIA monitoring in this anti-amyloid treatment world. How do we bring together clinical trials and clinical practice, different field strengths of scanners, different types of sequences, different sensitivities of these things to try to track that. And we'll also have, we'll look forward to seeing everyone at that, which they can still register for. We also have a number of immersives that are being offered on the Friday before the conference as well that are all sold out, that usually relate to things like imaging genetics, getting started with neuroimaging analysis, and a few featured research sessions that are being hosted throughout, like on sleep or genetics intersections as well, that we're often co-hosting with other PIAs.
Fantastic. Thanks, Sam. It sounds like you're going to be busy, and you'll be there, I assume?
I'll be there.
Right.
I'll be there from Saturday onward.
What about you, Ingrid? What's the sensory health and cognition PIA got going on at AAIC?
So given that we don't have a lot of time left, I really want to highlight one thing. We have an immersives on PIA Day, the Friday before the conference, and it's actually not sold out yet. So for anyone who has tried desperately to get into the neuroimaging PIA immersives, there's space left. Come to us, and it will be a lot of hands-on sensory testing that we will let people do. So if you want to know about your sense of smell, you can come Friday and
that sounds like fun.
Yes, I think it will be. Hands-on. So please come on Friday. If we're already at the conference, we'll be there starting from 7 a.m.
Wonderful. And there's have you got some? You mentioned a featured research session earlier as well.
Yeah, we submitted one, but we didn't pass. We will try again next year. Of course.
Absolutely.
Yeah.
Well, I went to I enjoyed watching Crufts this year as well, and watching the dogs do I have a wire-haired dachshund who is mostly useless, but I saw that they trained wire-haired dachshunds to be medical trained dogs who can sniff when somebody's having a seizure or their diabetes and things, and realize that my dog could be useful. So I think I'm excited by anything that involves going to a conference that involves having some kind of sniff test.
Perfect. I'm not sure you can bring dogs, though. I think there's a policy
No?
against that. I would love to meet him otherwise.
You can test it, but training dogs to do things is always good. Anyway, we go off topic. Thank you very much to the incredible Sam Lockhart and the wonderful Ingrid Ekstrom. I said that wrong again. But thank you both for joining us today. This live stream, as you all know, is part of the Dementia Researcher and iSTAART PIA Relay podcast series, which includes 6 episodes on our podcast as well. We've just released the 4th today. There's one more to come out tomorrow. Today's was we're talking about the Lewy body dementia peer with Joe Kane. We've got another one to come tomorrow, which is talking about the PIA, and then we're back to talking about the clinical trials and advancements peer, which comes out on our last one will be dropping on Saturday. And then we're also going to be recording a podcast every day during the AAIC to bring you highlights from a group of people who are attending virtually rather than in person. So this live stream again is part of that overall series, and you can find all of that series and profiles on both Sam and Ingrid and all of our guests across that on the Dementia Researcher website. And if you want to get involved in iSTAART, it's entirely free for students, and it's also free if you're based in a lower middle income country or in different parts of the world, and it's really inexpensive even if you're not. So you can get involved in the neuroimaging peer, the sensory health peer. There's a whole range of webinars and things go on throughout the year that make it well worth your while. So do consider joining those communities and do follow along with the series and join the rest of the live streams as we go. I think that's enough from me, but thank you again, Ingrid and Sam. Safe travels to London.
Thanks.
Thank you, Adam. Thank you, Ingrid.
And if you see them there, go look them up and say hi. Don't be shy. Thank you. Have a great time.
Yeah, come on over.
Bye-bye.
Thanks. Bye.
You have been listening to the Relay podcast, delivered as a collaboration between Dementia Researcher and iSTAART. This podcast is made at University College London with generous funding from the NIHR, Race Against Dementia, Alzheimer's Association, Alzheimer's Research UK, and the Alzheimer's Society. Please like and subscribe and share your thoughts in the comments.